Hello, I would like to thank the organizers of the NAPB meeting and this session for the opportunity to share a few highlights of our research in field-based high-throughput phenotyping over the past 10 years and convey our future research directions. The path from genotype to terminal phenotype, such as yield, covers a large range of spatial and temporal scales. The diverse set of processes operating at and across the intervening scales can have subtle effects on the terminal phenotype. However, they also govern a large set of endophenotypes or intermediate phenotypes, including properties like protein and metabolite levels, or physiological and morphological attributes of the plant that can be modeled. Furthermore, interactions between processes within and across scales can lead to emergent behavior. My talk today will focus on how we are merging the fields of plant biology and robotics to collect early season intermediate phenotypes, or endophenotypes, for building models that are highly predictive of terminal phenotypes at the end of season. Radical collaborations were essential to advancing DNA sequencing technology to where it now costs about $100 to resequence a human genome. This has resulted in a deluge of low-cost genomics data. In contrast, phenotyping is a 10,000-year-old challenge that started with the visual assessment of plants for desirable attributes at the time of plant domestication. Phenomics technologies have only just begun to evolve more rapidly, and phenomics data are typically more expensive to collect than genomics data on a large scale. This has caused a massive imbalance in the volume of data collected. Radical collaborations among plant biologists, engineers, and computer scientists are needed to bring the lab to the field, allowing multi-scale data to be collected. The concept of field-based high-throughput phenotyping, FB-HTP, has been advocated by those of us in the plant breeding community to reduce human effort, collect higher amounts of data, evaluate large population sizes at many time points in a growing season, decrease subjectivity when scoring plants, and diminish phenotyping costs over time. On the left is an example of a more classical physiological phenotyping approach with an instrument that collects highly detailed data but does not scale well to large plant populations. On the right is an example of a phenotyping system with greater throughput for non-contact, simultaneous measurements of multiple canopy phenotypes in the field environment. Stimulated by the need for better phenotyping in the field, I started collaborating in 2010 with agricultural engineers at the University of Arizona while I was a research geneticist working for the USDA ARS in Maricopa, Arizona. Our designed field-based high-throughput phenotyping system, as shown in this icon, was used to study the genetic basis of physiological traits and their relationship to lint yield in a cotton recombinant inbred lawn population. The field-based high-throughput phenotyping system could scan our field trial of nearly 500 plots in less than 1 hour. Furthermore, we use this rapid phenotyping platform to monitor changes in 3 physiological phenotypes at multiple time points within a day and multiple days across the growing season. And these 3 canopy traits were temperature, normalized difference, vegetation index, or NDVI, and height. With canopy temperature as an example, these data were used to map QTL expression patterns across cotton plant development. As an intermediate phenotype, these canopy temperature data were also found to have a moderately strong correlation with lint yield at peak flowering. Despite this progress, I was still driven by the need to better dissect the components of lint yield. In collaboration with Andrew French and Allison Thompson at USDA ARS Maricopa, Arizona, we demonstrated the potential of light detection and ranging, or LIDAR, sometimes called laser scanning, for phenotyping defoliated cotton plants. The field-based high-throughput phenotyping system, fitted with a LIDAR unit, scanned the entire field, allowing us to extract LIDAR data on a per plot basis and a per plant basis. In this point cloud visualization of a single extracted cotton plant, a Euclidean clustering approach was used to count the number of cotton bolls with reasonable accuracy. This was an initial indication to us that optical phenotyping could be used to better understand yield components. Ground vehicles and LIDAR units have evolved since our early work in cotton. In a collaboration with Ed Buckler, led by Joe Gage, a postdoc in his lab, the TerraSentia sub-canopy rover from EarthSense, fitted with a LIDAR unit, was used to characterize the whole plant architecture of maize plants in the field. Joe used the computational workflow to compress the 3D LIDAR point cloud data into 2D density maps, which were then analyzed with two dimensionality reduction techniques, principal component analysis and an autoencoder. The extracted latent space phenotypes, LSPs, were used to predict manually measured phenotypes by partial least squares regression. The LSPs contained enough plant architecture information to predict architectural and non-architectural traits. Joe is continuing this work to map G by time and growth curve parameters in maize hybrids that are part of the Genomes to Fields initiative. We now shift our attention from the ground to the sky. In the past few years, we have assessed the potential use of unoccupied aerial vehicles, UAVs, as efficient co-workers for precisely monitoring the incidence and severity of northern leaf blight, a maize foliar pathogen. The aerial system that we used at the time consisted, as shown here, of a DJI Matrice 600 and a Sony Alpha A6000 camera with a zoom lens. The large size NLB lesions as seen in the image on the right makes the disease an attractive candidate for image-based phenotyping. In a collaborative study involving Hod Lipson and his research group at Columbia University, and Rebecca Nelson and her research group at Cornell University, Harvey Wu, an undergraduate student in Hod's lab, used a convolutional neural network, or CNN, model pre-trained on ImageNet to transfer learn rather than training from scratch for a presence-absence detection of NLB lesions. The CNN model created interpretable heat maps, as shown here on the right, of the original images, showing the locations of putative lesions. The CNN model achieved 95% accuracy on a separate test set, and resolution was achieved at the sub-leaf scale. Beyond incidence, breeders and pathologists want to measure disease severity, which is a quantitative attribute. Thus, we attempted to estimate the number, size, and location of NLB lesions. Ethan Stewart, a former postdoc in my lab, trained a MASCAR CNN model to segment NLB disease lesions in UAV images. This deep learning model approach for instance segmentation accurately detected and segmented individual lesions in a holdout test set. The mean intersect over union, IOU, between the ground truth and predicted lesions was 0.73, with an average precision of 0.96 at an intersection over union threshold of 0.50. In these images shown here, the ground truth is marked with white polygons and predicted lesion instances marked in magenta, allowing you to visualize the high degree of intersection. One note is that due to variation in plant height and image resizing during dataset preparation, The scale here is variable between images and provided for general guidance only. I would like to now highlight our efforts in wheat and cassava. First in wheat. In a collaboration with CIMMYT, a former PhD student co-advised by Mark Sorrells and me, Margaret Krause, demonstrated the use of hyperspectral reflectance-derived relationship matrices for the prediction of grain yield in CIMMYT wheat in Mexico. The aerial hyperspectral phenotyping approach can monitor physiological changes resulting from plant growth stage and the environment. Therefore, hyperspectral reflectance-derived relationship matrices can capture both genetic manifests and genotype by environment interactions. This contrasts with pedigree and genomic relationship matrices, which are static. Shown in this figure are the single environment prediction accuracies of wheat yield across 4 site years, 2 environments in 2 breeding cycles, with G as the genomic relationship matrix, the A matrix derived from pedigree, and H as the hyperspectral derived relationship matrices. When considering the growth stage at the time of phenotyping, prediction accuracies were highest at the heading and grain fill stages. Overall, the hyperspectral-based accuracies are similar to or slightly higher than genomic and pedigree-based relationship matrices. I encourage you all to look at the results of multi-environmental prediction and correcting for days to heading in Margaret's G3 paper that was published in 2019. In our lab, we have also worked on the use of low-cost handheld spectrometers for phenotyping quality traits in plant breeding programs. Jenna Hirschberger, a recent PhD graduate from our lab, led a collaboration with IITA in Nigeria to predict cassava root dry matter with the handheld SIO spectrometer shown here on the left. Jenna found cassava root dry matter content to be highly predictable within 10 field trials. As shown in the figure on the right, mean partial least squares regression model performance range from an R-square of 0.62 to 0.89 for within trial predictions, which is within the range typically achieved with laboratory-grade spectrometers. A preprint at bioRxiv is available on Jenna's work if you would like to learn more. In general, technology has largely limited us to phenotyping macro-scale traits and indirectly measuring those traits at best. Working with Piyush Jian, a PhD student in Abe Strook's lab at Cornell University, and Wixin Liu, a former postdoc in my lab, and others at Cornell, we developed swelling shrinking nanoparticles with fluorescent reporters to rapidly detect water status inside a plant. Aquadust, as we call it, shrinks with decreasing water potential, and hence there is increased interaction between 2 fluorophores. We measure this interaction in terms of fret efficiency. Aquadust, which resides within the apoplastic region, allows for in situ, minimally invasive measurements of water status as water potential in local physiologically relevant microenvironments of leaves using an external sensor. This work represents a major step towards high-throughput phenotyping of water potential. Ultimately, we can couple water potential variation with biological and physical processes to accelerate our understanding of the factors that are critical for improving drought tolerance in crops. This work was recently published in PNAS this year. What is the next step for the use of aerial and ground systems in plant phenotyping? Through 2 recently funded NSF proposals, we are collaborating with plant biologists, engineers, and modelers at Cornell to collect phenotypic data with robots operating at the canopy, understory, and below-ground levels. Rob Shepard's robotics group will lead the development of a 1 to 2 foot worm-like robot that emulates how a borer drills into the ground, combined with a wave-like motion that copies how worms move through soil. By quantifying root features, soil properties, metabolites, and water, that includes the use of aquadust, we hope to use predictive models to combine the multi-scale below and above ground characteristics as intermediate features to predict such traits as grain yield and stress tolerance. Agriculture in the 20th century has relied on chemistry and correlative patterns. I think that agriculture in the 21st century will be the century of robotics and pinpointing the causal factors driving those correlative patterns. It will then better allow us to model what we know and, if needed, predict what we don't know. Our multi-institution group of collaborators comprising the proposed Center for Research on Programmable Plants, CROPS, is led by Susan McCouch at Cornell University. Notably, CROPS grew out of the Cornell Institute for Digital Agriculture, CIDA. Within the CROPS project, we share a vision of developing programmable plants with genome-encoded gene switches, sensors, and reporters with digital readouts serving as endophenotypes that can be read into software and acted upon by robots. We collectively call this the Internet of Living Things. How could we begin harnessing the genetic power of programmable plant populations? In one example, the CROPS Internet of Living Things could be used to perform intensive multi-scale measurements of endophenotypes across a training population of genetically diverse programmable plants. This will serve as the genetic framework for the use of genomic prediction models to predict endophenotypes such as protein abundance in developing plant tissues for hundreds to thousands of untested genotypes. It will further allow us to predict the function and activity of gene circuits and untested genetic backgrounds. I would like to thank all those that have contributed to this work and the funding agencies that have provided support, especially Columbia University, University of Illinois Urbana-Champaign, EarthSense, University of Arizona, NextGen Cassava, USA LARC, and all of our collaborators at Cornell University. I'm happy to take your questions at this time. Thank you very much for listening.